Before the end of the 1950s, an extensive investigation which laid the foundations for the concept which is used currently for the study of membranous digestion. In 1960 to 61, it was found that the rate of enzymatic hydrolysis of starch during the perfusion of intestinal slices of a white rat or during incubation of inverted section was several times smaller than the rate of hydrolysis of starch at the moment of its passage across the intestine. Simultaneously, at the end of the 1950s and at the beginning of the 1960s, two different hypotheses to explain these facts were presented, namely number one, the concept of apical intracellular digestion, and two, that of the membranous digestion. Evidence is increasing to suggest that the first hypothesis is unattainable. Okay, so what is membranous digestion? It's the process of enzymatic splitting of nutrients by enzymes located in the glycocalyx and apical membranes of enterocytes. These enzymes include alkaline phosphatase and aminopeptidases. Membrane digestion is due to enzymes absorbed from chyme and enzymes structurally associated with the membrane and intestinal cells. Of particular significance are the electron microscope studies of Oda, Secchi, and Johnston, and others who have also demonstrated the presence of globules of 50 to 60 A in diameter at the external membrane surface using the negative contrast of the brush border. These globules were solubilized and turned out to be at least the barriers of disaccharide activities, and protease is also shown. It is very likely that other enzymes such as pancreatic DNase and RNase may be absorbed and participate in membrane digestion. Indeed, it was the first to pay attention to the importance of these structures in the process of membranous digestion. In addition to that, in the early 20th century, the Russian physiologist Ivan Pavlov conducted research on the digestive system, particularly on the process of digestion in the stomach and intestine. One of his colleagues, Nikolai Ugolov, expanded on this research and made significant contributions to our understanding of membranous digestion. Membranous digestion refers to the breakdown of food within the walls of the intestine rather than in the lumen, the open space within the intestine. Ugolov discovered that the process is facilitated by enzymes that are embedded in the membranes of the intestinal cells. These enzymes are able to break down complex carbohydrates and proteins in smaller molecules that can then be absorbed by the body. Ugolov's research also showed that the process of membranous digestion is regulated by a number of factors, including the acidity of the stomach, the presence of certain hormones, and the composition of the diet.
Membrane digestion is any other catalysis on the surface depends on the rate at which substances pass from liquid medium onto the surface, and under normal conditions that depends upon intestinal motility. Thanks to the continuous elimination of the intermediate products of hydrolysis into the membrane digestion zone, digestion in the cavity is intensified. The enterocytes are highly catalyzed columnar cells with usual organelles such as endoplasmic reticulum, Golgi complex, and numerous mitochondria. On the apical surface of the cell, we have microvilli and glycocalyx, which collectively make up the brush border with enteric sheath enzymes such as dimer, and absorbed enzymes which are called supplements. Microvilli are projections that help increase the absorptive area of the cell, while glycocalyx are thin filaments layer of mucopolysaccharides of material houses, important enzymes that function in the membranous digestion process. Thus, membrane digestion is one of the three basic stages in the activity of the alimentary tract. It accepts the products of intermediate hydrolysis from the cavity, by which all the cavital processes are intensified. It accomplishes also the hydrolysis of 70 to 80% of the primary molecular bindings in the food by polymers. It couples the digestive processes proper and those of transport. As food passes from the small intestines, the enterocytes facilitate the uptake of important substances such as glucose, amino acids, fatty acids, minerals into the bloodstream. Apart from that, they also contribute to the maintenance of the intestinal barrier function, preventing harmful substances and pathogens from entering the body.
It is very important to study the disorders of membrane digestion because of the etiology of a number of its diseases and for promoting progress in diagnostics and therapeutic research. Enzyme absorption disorders. Maciejewicz, Zawelensky, and Ugolev have developed the fraction technique of investigating enzyme absorption. Recently, changes in lipase absorption have been seen in patients suffering from atherosclerosis when compared with healthy controlled diseases as enteritis, enterocolitis, and diseases of the biliary duct. There is damage to the villous structure and disturbances in the epithelial elements in proteosynthesis. It is of interest that disorders of the membrane digestion are likely to appear earlier that can be seen by histological and histochemical changes. Synthesis and migration of enteric enzyme in disease study of genetic and acquired defects of the enzymatic properties of enterocytes shed light upon a great number of diseases of the digestive system. Many enzymatic defects remain beyond the understanding of both the laboratory investigator and the clinician. It is important to remember that in the same disease, certain enzymatic defects can depend on disorders of synthesis and other on disturbances of transfer. For example, in severe X-ray injuries, hydrolysis of both disaccharides and peptides is completely stopped, but the absence of invertase activity depends on the suppression of the synthesis, whereas peptidase is synthesized in amounts close to normal, but it is not included in the membrane of the microvillus. It is interesting that under the influence of different stresses, disorders of the enterocyte digestive function depends rather on disturbances of transfer than on enteric enzyme synthesis. Other defects of membrane digestion. When hydrolysis in the digestive cavity is slowed as a result of deficiency of the basic digestive glands, membrane digestion is impaired due to an insufficient supply of substrate into the brush border. This is accompanied by the enzymatic and transport function of distal portions of small intestines being greatly enhanced, a situation which is defined by both as a reserved zone. The total volume of membrane hydrolysis can diminish as a result of a reduction in the epithelial population in the small intestine, first, due to its shortening, as in surgical operations, and secondly, to smaller numbers of enterocytes in the VD. It should be noted, however, that in many cases, membrane digestion is a process compensating for the defect of cavidal digestion. This was demonstrated in particular in connection with the assimilation of polysaccharides when the external secretory function of the pancreas was excluded. To sum up, the actual functioning of the digestive apparatus in higher animals and man comprises of three successive and interconnected stages. First, the cavidal digestion, second, the membrane digestion, and lastly, absorption. The study of membrane digestion is not only of theoretical interest but of practical importance as well.
For the immobilization of the rate, we used intraperitoneal anesthesia injection. We injected several rates, about 3 of them.
Okay, so in this procedure, we are going to cut open the abdominal cavity of the rat. Here we are cutting it open. Now we are going to remove the other part of the small intestine in order for us to locate the proximal part of the small intestine. Okay, so we have gotten the proximal part of the small intestine and we are to cut it. We are supposed to extract a small piece about 3 to 5 centimeters long. Okay, the small intestine has been successfully extracted and now it is placed in a dish with water fully immersed.
Using glass rods with a small club-shaped extension at the end, the extracted intestinal wall or area is inverted by the mucous membrane to the outside. This inverted mucous membrane is then washed in a physiological solution for 30 seconds in order to remove enzymes from the surface of the intestine and the intermicrovilli space. Enzymes providing cavity digestion.
Place 2 test tubes on a rack, one with an experiment with 0.1% of starch and the other control with 0.1% starch. And then using a tweezer, pick up the inverted small intestine and place it to the experiment test tube. To make sure that it has been fully immersed in the physiological solution, we use a glass rod.
So we have the two test tubes, one control and one experiment. In the thermostat, it has already started to be incubated for 30 minutes at 37 degrees Celsius. One of the test tube contains 1 milliliter of 0.1 solution of starch, while the other test tube contains 2 milliliters of Renga solution. So we're going to keep them there for 30 minutes. We're going to close back the thermostat and put it at 37 degrees Celsius as shown in the thermostat.
At the end of incubation, the test tubes are removed and put onto a rack. Into each test tube, we add 1 to 2 drops of Lugol solution, and then we shake these test tubes.
After we observe more intense color in the control tube comparatively to tube with piece of intestine, this is due to the presence of starch in the control.